Deutetrabenazine is a VMAT2 inhibitor indicated for the treatment of chorea associated with Huntington's disease. Deutetrabenazine, which had orphan drug status, is a deuterated form of the VMAT2 inhibitor tetrabenazine. It's the first deuterated product approved by the FDA and only the second product approved for Huntington's disease. The molecular weight of deutetrabenazine is 323.46 and the pKa is 6.31. It has a molecular formula of the following. Deutetrabenazine is a race-mixed mixture containing the following structures. Deutetrabenazine is a white to slightly yellow crystalline powder that is sparingly soluble in water and soluble in ethanol, which is an isotopic isomer of tetrabenazine. The mechanism by which deutetrabenazine exerts its anti-chorea effects is related to its action as a reversible depleter of monoamines from nerve terminals. The major circulating metabolites of deutetrabenazine are reversible inhibitors of VMAT2, resulting in decreased uptake of monoamines into synaptic vesicles and depletion of monoamine stores. It also regulates the levels of neurotransmitter dopamine. The FDA's approval of the drug was based on a single study which was randomized, double-blinded, placebo-controlled parallel group study. It was conducted with 123 patients screened and 90 admitted to the course between 2013 and 14. In total, 87 patients finished the trial period. The deutetrabenazine treatment group mean age was 55.4 with an equal number of men and women, with the placebo treatment group mean age being 52.1 with 28 men and 17 women. Drug administration lasted 8 weeks with 4 weeks of maintenance with a 1-week washout evaluation at the end. Dosage started at 6 mg per day and increased weekly by 6 mg per day until chorea was adequately controlled. Mean deutetrabenazine dosage was 39.7 mg per day and 43.3 mg per day in the placebo group. The study used 2 primary objectives, the first being clinical evidence in reduction of chorea and the second objective being for the quality of life improvements. Primary outcomes found were individuals in the deutetrabenazine treatment group had mean negative 4.4 improvement in chorea score, with endpoint being the change in total maximal chorea score from baseline to maintenance therapy. Individuals in the placebo group had an improvement of negative 1.9. Chorea score ranges from 0 to 28, showing the lower the score, less the effect of chorea on the patient, with 7 body regions analysed. The chorea scale is graded on a 0 to 4 scale, with 0 showing absence signs and 4 showing marked and prolonged signs of chorea. Following the washout, chorea levels were returned to baseline levels in both groups. Secondary outcomes were, with 42% of patients in the deutetrabenazine group reported successful treatment using the CGIC scale, with 51% reporting success in the PGIC scale, both of which are 7-point Likert scales and are subjective. In comparison, the placebo group had 6 patients and 9 patients report success respectively. The primary side effect found was somnolence, but didn't need a drug dosage reduction along with diarrhea. Dosage reduction was needed in 3 patients in each group, with adverse effects common in both groups. A patient experienced cholecystis and agitated depression in the deutetrabenazine group, whereas in the placebo group, 1 patient experienced an obstructive pulmonary disease aspiration event. In both cases, the drugs were suspended. Depression is a concern in any patient with Huntington's disease, with an increased need to monitor patients taking VMAT2 inhibitors. This is important since the drug targets the VMAT2, a transport protein, on presynaptic neurons to help reduce the involuntary writhing associated with the disorder. Currently, tetrabenazine is the first drug of choice in North America and Australia used for reducing chorea. Tetrabenazine is well tolerated and effective. A study of tetrabenazine patients over a period of 3 years showed the overall effectiveness on chorea declined despite dosage increase. Similar studies also indicated dose-related adverse effects and increased risk of depression and suicide ideation, making this a serious concern. This drug requires less frequent dosing due to longer half-life, a lower daily dose, and improved tolerability. Studies showed no significant difference from tetrabenazine in efficacy or adverse events, indicated by the results from tested deutetrabenazine against placebo. To learn if deutetrabenazine is more effective and safe in treating chorea in Huntington's over tetrabenazine, a direct head-to-head comparison study is required. An indirect tolerability comparison has been made from various clinical studies on these 2 drugs tested against placebo, which suggests that in treatment of chorea, deutetrabenazine has a favorable tolerability profile compared to tetrabenazine. 4 studies addressed the patient's perception of treatment success. A phase 3 trial directly compared patients switched over from tetrabenazine to deutetrabenazine. Patients with chorea were given tetrabenazine for 8 weeks prior to switching overnight to deutetrabenazine. Results indicated chorea control was well maintained after 1 week and significant improvement at week 8 with low rates of neuropsychiatric side effects. Concluded that patients can switch drugs with favorable safety and tolerability without loss of control over their chorea. Another study evaluated the efficacy, safety, and tolerability of deutetrabenazine fixed doses, which showed that after 12 weeks at 24 and 36 milligrams per day, this significantly improved AIMS scores by negative 3.2 and 3.3 points respectively, with only negative 1.85 points to the placebo group. The drug had a favourable safety and tolerability at fixed doses, and that an effective treatment can be individualised for patients according to the tolerability and control of chorea. Another study analysed the long-term safety and efficacy in patients with tardive dyskinesia. Patients showed improvement in AIMS scores scoring negative 5.1 points from baseline, as well as SIGIC scale, which demonstrated 72% of the patients quantified change as much improved. Deutetrabenazine at 54 weeks of treatment was safe and well tolerated by the patients with low rates of adverse events. The final study was conducted for the effect of deutetrabenazine on quality of life for tardive dyskinesia patients. This was assessed using the MCDQ-24 subdomains, which showed improvement at week 12 in daily activities and stigma on treatment with deutetrabenazine at 36 milligrams per day when compared to placebo, indicating a clinically significant improvement in life. These studies corroborate with earlier studies that indicated that deutetrabenazine has a favourable safety and tolerability profile compared to tetrabenazine and outperforms placebo for the treatment of chorea and also improved quality of life. In addition, patients were more likely to perceive treatment success with the drug compared with placebo. Overall, deutetrabenazine moves towards better treatment of Huntington's chorea while reducing side effects at the same time.